All right, we're going to go ahead and get started. Can everyone hear me all right? Microphone's good. All right, well, welcome to BIMM 100, Molecular Biology. My name is Amy Pasquinelli. I will be your instructor for this quarter. Hopefully you all are successfully registered. You are able to access the Canvas site and saw all the useful information that is posted there already. I highly, highly recommend that you read this course details. I know it's many pages long, but hopefully it really provides you with all the information you're going to need to know how this course is going to run and what the expectations are. I'm just going to go over a few key points in class today to highlight a few things. This is how to get in contact with me. The best way to get in contact with me is here in class. So I'll try to come early to class, come talk to me beforehand. I almost always can stay after class. If you have questions or anything, come talk to me in person then. I will be holding an office hours every week. This will be starting next Thursday. Come visit me. I'd love to meet all of you in my office hours at some point. You all know where the website is for this class on Canvas. You're here, so you know where and when the classes are going to be held. If you miss a class, if you are sick, don't come to class. If you think you might be a little sick, wear a mask. We really appreciate that. It's really hard to learn or teach when you're sick, so let's really look out for each other and all try to stay healthy. Your grade is not going to be affected. You're not going to miss out if you miss a class or two. You can catch up on everything on the podcast, and these will also be available on Canvas in the media gallery. These dates are very, very important. Put them on your schedule now. This is when you will have graded exams, these pre-midterm and final, as well as the in-class or the register scheduled final. And as we get closer to those exams, I'll tell you more about what to expect. All right, you are very fortunate to have an excellent team of instructional assistants who will be aiding with this class. Can you please stand up so you can see what they look like now compared to their pictures that might have been taken a few years ago? So you have your grad IA Pierce, and then Hamangi, as well as Tessa, were actually two students in my class last year. Take advantage of their knowledge of how this class runs, of how they excelled in molecular biology last year. And so they will be running the discussion sections, which will be starting next week. Next Friday actually will be the first set. And office hours will actually begin next week, beginning on Monday. Their schedule, you guys can sit down, thank you. The schedule for the office hours has been posted on the Canvas website, so even though you might not have any homework yet, if you want to meet them at their office hours, I recommend you go check that out. All right, so we're going to use clickers in this class. Probably many of you have used clickers before, either the device or you can have the program on your phone as well. This I really like to use in my class because it's a way to get feedback on how well you understand a concept I just taught. So I am learning from you how well I taught something, and in return, you can get extra credit points for helping me with this. To get those extra credit points, you need to actually participate in a poll in the class. So you can't just come to class, keep your clicker in your backpack. That is not going to count. You actually need to click in for the polls that we will run in this class. There will be 19 classes that we will have clickers used in. You can get up to 10 points of extra credit. So what that means, if you click in a class, you get 1 point. Next class, 2 points. If you forget your clicker or your battery dies or you're not there in class, doesn't matter. You have so many other opportunities to get this maximum of 10 points extra credit that will be added to your grade at the end. If you've never used a clicker before, I have posted instructions on the Canvas website for how to set it up to make sure it's working well for you. The clicker questions that I show during class, I will post them at the end of the week for every class. So you don't have to worry about taking pictures while they're up there. I will post those questions after we've done them for that week. The frequency in this class is BC. Make sure you are on BC or your clicking in may not be registered. Okay, so the exams. This is a whole lot of detail, and hopefully you have already read it in the course details. As I said, when we get closer to the midterm and the pre-midterm exam, I will talk more about what to expect for those. But the bottom line is the midterm exam is going to be in class during normal class time. The pre-midterm as well as the pre-finals, those are going to be Canvas quizzes that will be open for a 48-hour period of time, when you can choose that you have the best time to take either an hour and 20 minutes or 3 hours to sit down and take that Canvas quiz. Another thing to be aware of for these pre-midterms and pre-final exams, you only need to get 50% correct to get a full score on it, right? So these really are there as an opportunity for you to practice what you're going to see on the real exam, to get a good idea of where your deficiencies are so you know where to focus your studies. And again, we'll talk more about them as they come up, but don't forget to take them. Put them on your calendar right now. Make sure you have a window during those dates. They are listed on the syllabus when you will be able to take these exams. All right, the other part of your grade, and it is only worth 1 point, but you absolutely must do this or I will be very suspicious of you. So you'll see that already under assignments, I posted this integrity pledge and class commitment. So please do that, submit it. It basically confirms that you are going to use integrity. You are not going to cheat as you take this class. It also says that you have read the course details and understand the expectations. So before you sign this, please read those course details. And then you can type your name if it's too hard to get your signature in there, that's fine. And then the other half point you'll get for turning this in is I want you each to have 2 thoughtful commitments that you're going to make to yourself to help you excel in this class. We all learn differently. We all have different styles of learning, but think about what works best for you, or maybe what hasn't worked for you in the past, and you're going to try in this class this quarter to really help you do better. And so I want you to write those down as well. I think about 5% of you already saw this and already submitted it, so that's awesome. It is due next Thursday, I think at 5 o'clock, so hopefully this pie chart will turn all green by class next week. Very simple, think about it, and then turn it in. And along with this commitment you are making, let me just be very, very clear. Do not cheat in this class. You will get caught. Every year we catch people cheating. It's very sad, but I make no exceptions. You will be immediately reported to the Office of Academic Integrity. You will almost definitely fail this class and perhaps have more significant punishment with that office. Over 1,000 of you get reported to the Office of Academic Integrity every year with a huge boost during COVID. I know it was a hard time. Things have been getting much better after COVID, but this past year the rates are going back up. So please, please, please do not cheat. It is not going to help you in this course. You're going to get caught. So let's make sure we're all on the same page. Take out your clickers. Here's a clicker opportunity. You can get 1 point extra credit right now. So make sure you are on BC and click in which of the following are considered violations of academic integrity and would result in a report to the Academic Integrity Office, along with loss of points and possible failure of this class.
Yeah, that is true. You can't narrow down into very big data. Yeah. Darn. Somebody should tell the administration. Wait, shoot.
Oh yeah, you did change your style. You were right.
I told you my hair was going to be a different color by Thursday.
That's why I had to have you all stand. And your picture looks very different too. You had a different hairstyle. And your
I just put product in today. They generally
You just tried today?
yes, they try to look.
Everybody keeps.
All right, 10 more seconds. Put your final answer in. So most of you brought your clickers today. That's great. 170 of you. Awesome. All right. Okay, so we have a very, very strong consensus for E, all of the above. Although a few of you think maybe it's only B or maybe it is only D. So let's make sure we are all on the same page. A, is this cheating, posting, accessing, or any method of giving or getting answers to an exam or assignment? Yes, absolutely, this is cheating. There will be times when I tell you, you can work together, you can use your book or things like that. If I give you explicit instructions, you can use those instructions, and that is all fair game, right? So what about copying answers from the internet or some sort of AI agent or previous year answer keys or a person? That is cheating, yes, absolutely. If you're turning in something that you said was your own, but it really wasn't your own, that is cheating, okay? So B is correct, but also all of these are correct. So C, having someone else use your iClicker to get extra credit. This one gives me so much pain because it's really just extra credit and you have multiple opportunities to come to class and get that extra credit. We have had this happen multiple times in the past. They are caught. Sometimes they've been turned in by their peers, and they lose all their extra credit points, and they also get reported to the Office of Academic Integrity. Don't do that, okay? Don't do that. D, having someone else or something else take your exam, all right? Absolutely not. When you take your exams in person in this class, you will have to bring a picture ID. We will verify that it is you handing in your exam and that you took it, all right? So don't even get thoughts about hiring people or doing anything strange like that. So absolutely all of the above, and there's many more examples I'm sure we could come up with and think about. The other thing I want to say is if you are unsure about something, just ask me. A few years ago, there was an issue with some students being emailed, hey, let's work together on the final exam together. We can do this. And the students who got the email were so upset because they're like, oh my gosh, I'm going to be accused of cheating. And luckily, one of them came to me, and I'm like, no, you didn't do anything wrong. Someone approached you, you reported to me. And then the other students who that had happened to were so relieved. So don't worry. Just come talk to me if something's suspicious or if you have a question, or you'd be like, am I allowed to use that to do the homework or not? Or am I allowed to use that for the pre-exam? Just ask me, okay? All right, so moving on. The other points that are normal points that you need to earn for this class are by going to discussion sections and participating. We are not taking roll as you go to discussion section. We are only recording you if you participate by asking a useful question or answering a question. So useful question, something that would be unuseful is like, is this going to be on the exam? That doesn't help with the discussion section. So there will be 8 different discussion sections throughout the quarter. You need to participate in 4 of them to get a maximum of 4 points. So again, if you miss a discussion section, you're sick, it's not going to matter. Just make sure that you get those points and get them early on in case something does happen throughout the quarter. The discussion sections, it's going to be easy to participate in them because they are going to entirely revolve around the homework that you have been assigned. So almost every Thursday, not tonight, but starting next Thursday, I will post a homework assignment for you to practice the material that we have learned that week. That will be the basis of the discussion sections. Your IAs will be going over that homework during your discussion section. So anything that you were confused about on the homework, it's a great time to get clarification. Anything you felt really confident you got the answer, volunteer and volunteer that answer. Make sure you say your name, and then you'll be recorded, and you'll get those points. The other great thing about homework is it is yet another way to get extra credit in this course. So if you do the homework and you submit it before the very first discussion section starts, you can get 1 full point of extra credit. So the very first discussion section starts at 10 a.m. every Friday, which means you need to submit your completed homework before 10 a.m. that Friday. If you don't, you didn't have time to do it, but you're going to do it later, or maybe you want to do it while you're in discussion, you can do that. And then as long as you submit it before noon on Tuesday the following week, you can get a half point of extra credit. The reason for that is at noon on Tuesday, I will then post the answer key, at which point you cannot turn in the homework for extra credit anymore. So do this. Again, there's a maximum amount of points you can get, a maximum of 5 points. So if you forget to do it 1 week, or you only have time to submit it for a half point 1 week, that's fine. You still have a lot of opportunities to gain extra credit, potentially up to 5 points for doing the homework and submitting it. As we just talked about, clickers, you can get extra credit that way, up to 10 points, and then also outstanding participation in your discussion section or in this class itself can earn you a point or 2. So what that breaks down to is here are all the scores that you can get. Remember the pre and the 2 pre-exams, even though the pre-midterm has a total of 40 points possible, you only need to get 20 points to get a perfect score for that. Same idea for the pre-final, 80 points will be possible, but you only need to get 40 of those correct to get a full point. And then that adds up to 185 points, and there's up to 17 additional extra credit points, which we just talked about here. Does anyone have any questions about sort of the grading for the class and the format that we have talked about so far? Yes?
Do you need to attend the discussion section on the website you signed up for? Or could it be for any one of the 4?
You need to attend the one that you signed up for because there's limited space for students in the classrooms to which it has been assigned to. So if you have a conflict, I think what you probably need to do is remove yourself from the class and then re-sign up for the discussion section that works for you. Yes? Can you bring cheat sheets? Let's not call them cheat sheets. For the in-class final and midterm, no. You can use no materials. But if you read the course details, you'll see that for the Canvas quiz, you are allowed to use your notes, your textbook, things that will help you with that. Other questions? Yes?
Are exams entirely on lecture content?
Great question. The only thing you will be tested on for your exams is what I have presented in class. If you are using the textbook and you read something in the textbook that we never talk about in class, you're not going to see it on an exam. The exams will revolve around what I present in class as question formatted in the clicker questions and the homework that you will be getting. Other questions? All right, so that brings us then to the syllabus, which hopefully you also were able to access. And so here we are today at lecture 1, and so I'm giving you an overview of the class, and then we'll talk a little bit about DNA and DNA structure. There's no homework assigned tonight. If you are using the textbook, those are the pages. The textbook is optional. Some students find it very helpful to read ahead of time the material that we're going to go over in class. Other students have told me that it's confusing, so it's entirely up to you. And like the question that was just asked, if you read something in the textbook and I never talk about it, you are never going to see that on the exam. And so you can pretty much follow this. You will see this will be the last material we will have before the pre-midterm and the midterm. The pre-midterm will probably open up right after class for about a 48-hour period for you to take that Canvas quiz, and then we'll all meet here on Tuesday and take the real midterm. Whoops. And then the rest of the schedule, also it's pretty self-explanatory. We have a few days where it's holidays, so we will not be meeting here. And then finally, the registrar has scheduled us for our final exam in this class on Thursday of finals week. And again, the pre-final will be after the last class for a 48-hour period. All right, any questions about the syllabus? All right, so questions for you. Why are you taking this class? You're dedicating a lot of time and effort to being here, so why are you taking this class? You want to have intention for everything you do, right? So why are you here? Yes? To graduate. Okay, so, but why do you need this to graduate? It's required for your degree. What's your degree in? Human bio. So what do you want to, why do you want to be a human biologist? Yeah. So most med school track people are going to have to take molecular biology. Who's pre-med? Even if you're maybe pre-med, maybe, yeah, still maybe. Right, so a good number of you are pre-med, so this is probably required for your major. Hopefully it'll help you for your MCATs. It'll help you for actually being excellent doctors in the future. Why else are others of you taking this class? Yes? Yeah. So all the molecular mechanisms and pharmaceuticals, why we take certain drugs and how they affect our body. Are you thinking of pharmacy school? Pharmacy medical, yeah. So pharmacy, who wants to go to pharmacy school? Yeah, we need a lot of good pharmacists. Okay, yeah, not a whole lot, but we need pharmacists, definitely. What else? Why else are people taking this class? Yes? Yeah. So definitely, genetics used to be a prerequisite for this class, but it no longer is. So we definitely talk about genetics, or the basis of genetics, which is DNA and the molecules underlying it. What do you want to do when you graduate? Med school? So we have med school, we have pharmacy school. What do the rest of you guys want to do after you fulfill your requirement by taking this class and graduate? Yes? Yeah, research. So maybe get a master's degree or perhaps a PhD or go into industry. Who wants to do something like that? Be involved in, biomedical research, okay? A lot of us for that. Good. So this is a very foundational class for that. Yes? Yes, you. Yeah, so into industry, yeah. So, dentistry, sorry. Yeah, yes, dentistry. We need good dentists, right? Yeah, so who else wants to go to dental school? Yeah, good. Yeah, interesting. Yeah, so this is very important and probably part of your major and will help you get into dental school. What are we missing? Yes, way in the back. Biotech, yeah. So biotechnology, industry, pharmaceutical companies, all of these need well-trained scientists to make the next big discoveries that are going to help us live longer, healthier, cure diseases, deal with climate change, all the things that science can really contribute to. Am I missing anything? How about nurses? Who wants to go to nursing school? Oh, no, we need nurses. Oh, my gosh. All right. So, yeah, nursing school. This is another prerequisite and something important for that. Yes? Vet school. Who wants to go to vet school? Yeah, help our pets, help our animals. Right. You want to be a well-educated veterinarian. Anything else? I think the most original answer I had to this question several years ago is I had a student who told me he wanted to be a screenwriter for science fiction movies, and he wanted them to be more science than fiction, and so hopefully this class helped him with that. All right, so those are some good reasons why you're taking the class, and think about this opportunity to learn as much as you can to help for your future careers and what you want to do next. Then we have to come to this question of, so what is molecular biology? Okay, we kind of know why we're here, but what is this class and what are we learning? And so one very famous person, who knows who this is? Who recognizes this? He's no longer with us, but maybe there's a hint here. Crick, yes, Francis Crick of the famous Watson and Crick. What did they do? They solved the structure of DNA. We'll talk a little bit more about in this class. And so in addition to that, he is one of the founding fathers of molecular biology. And understanding the molecules and the processes they're involved in. But I like this quote from him. It says, molecular biology can be defined as anything that interests molecular biologists. So that's kind of a license for me to teach you anything, right? But promise I won't do that. I will stick to the standard curriculum that is critical for understanding the basics of molecular biology. So we need a better definition. And in fact, if you have the book and you read this entire book as I have, you will not find a single definition of molecular biology in it, surprisingly. So if you go to the Encyclopedia Britannica, it actually gives you a pretty good definition. And so this resource says that molecular biology is the field of science concerned with studying the chemical structures and processes of biological phenomena that involve the basic units of life, which are molecules. The field of molecular biology is especially focused on nucleic acids, which are going to include your DNA and RNA, as well as proteins, macromolecules that are essential to life processes, and how these molecules interact and behave within cells. So that's big picture what molecular biology is. But maybe even a better question is to start to think, well, what do you want to learn in here? So, we heard some general ideas. We want to learn molecular underpinnings. Does anyone have something specific they want to learn in this class? Yes?
I want to learn about the bacterial interactions.
Bacterial interactions for DNA. You mean like how DNA is replicated in bacteria cells or even in our own cells? Yeah, well, we will talk about some prokaryotic mechanisms, although largely we'll be talking eukaryote, but the general process is so well concerned from every life form does DNA replication, pretty much the same with transcription and same with translation. What else? Yes? Yeah, so that's a really big part of what we're going to talk about in this class is gene regulation. How does a particular gene turn on in your correct cell type at the right time, to the right level? How does it get shut off? We're going to definitely be talking a lot about that at the transcriptional as well as post-transcriptional levels. Yes? Yeah. So once we have gone through all the molecular mechanisms, the last class that we will have that will have new material, we'll be putting together what we learned to think about the process of cancer, how genes change and become misregulated and cause cells to divide out of control, which is essentially what cancer is. Yes? You want to learn about viruses, and yeah, so this is another class we'll have right at the very end because we'll take what we have learned about basic molecular mechanisms, talk about how viruses take over our cells to use those molecular mechanisms for their own evil purposes. Good. Anything else? Good, so we did cover some of the things, some of the questions that have come up in past years that have been really of general interest, and so the first one is directly related to the suggestion we had in the front here. And so if you think about it, your body is composed of all these different cell types, all these different tissues. They all have the same DNA in them. How can they all look and function so differently, right? Your heart and brain cells, so these are heart cells, these are brain cells here, they look really different and they perform really different functions. How is that possible? So it's gene regulation, right? Turning on the right genes, keeping the right genes off in those different cell types. And we'll spend a good amount of time in the next few weeks talking about gene regulation. We'll also talk about a number of different molecular methods. One of the most common and probably one you've all heard about, PCR. What does PCR stand for? Polymerase chain reaction, right? If you've watched any like CSI or crime investigator, they're always going to do a PCR to identify the criminal or to rule people out. And so this is a method who's used it in a laboratory before, in a lab class or in their own lab? Yeah, so everyone's probably done it, but maybe we don't all know actually how it works, what those primers are actually doing, how you're amplifying DNA. So we will learn about that, how PCR is used in forensics, medicine, as well as in general laboratory use. If you have diabetes or you know someone who has diabetes, you have bacteria to thank for all that insulin that helps keep you healthy. We have learned how to turn bacteria into little machines, little generators to pump out human proteins like insulin. And so we'll learn in this class about how bacteria can be engineered to synthesize human proteins that are very medically and many examples now environmentally relevant. We heard an interest in viruses, right? So especially these days when viruses have impacted, or one particular virus has impacted our life so much over the past few years. And so we will talk about how these viruses are able to get into cells, how there's many different types of viruses, they have many different genomes, what they need from their host cell, and how they use that to propagate and make more of themselves. We are now at a point where we have amazing methods at our fingertips. In fact, at this point, we can design therapeutics to inhibit the expression of any gene we're interested, at least in the laboratory, right? And so does anyone know, I'm thinking in particular of two different methods that you can, if you know the sequence of a gene, you can knock it out or knock out its expression. Does anyone know, have an idea of what one of those might be? Sure. So CRISPR, many of you hopefully have heard of CRISPR, so this ability to edit genomes to change the DNA sequence. You can change the DNA sequence to stop it from making a protein. CRISPR is also amazing because you can not only knock out or mutate a gene, but you can also fix the sequence because you can recode the DNA to be anything you want. And in the laboratory, we do this all the time now. It's very possible, and there's a lot of course, ethical implications for that in using this in medicine and human patients, and we'll talk quite a bit about that. There's another method that we will talk about that is more knocking out expression of any gene. Again, you only have to know the sequence of it, and you can deliver very simple molecules and cause it to no longer be expressed. Does anyone know what method I'm thinking of? RNA interference, exactly. And so we'll also talk about this method and how readily you can knock out the expression of a viral protein so that virus cannot replicate. These work great in the lab, but we still need industry and biopharma to figure out how to make these all medically possible. We're now in the genomics era, so today, I could take one of your skin cells and sequence your DNA in 24 hours and have the entire 3 billion base pairs of DNA that encodes for you. This is amazing, right? And so this is the basis of what's now called personalized medicine, having your entire DNA sequence and seeing, do you have certain susceptibilities to different diseases, and trying to understand how that can relate to maybe medical treatment that you might need. And so we'll learn about how DNA at this level can be sequenced and also how this is being used for personalized medicine. And then finally, we mentioned an interest in cancer, and indeed in the last class, we will talk about how carcinogens can cause mutations in your DNA. And if you get those mutations in the wrong genes, that causes them to be lost or overexpressed, and that promotes the cells to divide out of control. That's what then leads to cancer. And so this is just sort of an idea of the various questions and themes that we'll be talking about throughout the quarter. All right, any questions before we get started with sort of more lecture material? All right, so we're talking about, yes, question in the back.
How does it feel working in a lab that won Nobel Prize?
He's asking me how it feels to work in the lab that won the Nobel Prize. We can talk about that when we talk about the microRNA lecture, which is, so last year the professor that I did my postdoc with won the Nobel Prize. And I actually got to go to Stockholm with him to celebrate that. So it was amazing. We can talk more about it in office hours or sometime off of the podcast. All right, so I guess it was before Nobel Prizes were a thing. We have Charles Darwin. This is sort of where we consider molecular biology to be initiated. Because Darwin came up, of course, with this theory of evolution. So what is the theory of evolution? What is this? So what does it mean? How does evolution work? Yes, in the middle back there. Exactly, survival of the fittest, right? Environment changes, those who do better in that environment are going to survive, pass on their genes, and propagate. Right, and so when we talk about, the theory of evolution, it's really survival of the fittest. Those are changes at the genetic level, changes at the DNA level that are causing different gene expression, and if that makes the organism fit better for their environment, their circumstances, they're going to propagate. There's an example of evolution that has been happening right before our very eyes very rapidly in the past 5 years, let's say. Who knows what I'm talking about? Yes? Yeah, so bacterial resistance, and that's been happening more like over the past 20 or 30 years, absolutely. So, and a lot of that can be attributed to if you don't take your whole course of antibiotics, and so not all the bacteria die, and those that pick up a mutation and can deal a little bit better with that antibiotic, they will propagate, and then they can become resistant. So bacteria, and then what is the other? Yes? Yes, all these COVID variants we have to deal with each year. So a variant is just a subtype of a virus that is mutated, making it different from its predecessor. When a virus is infecting your cell and propagating, it's making billions of progeny, and it's making mistakes, and there's a lot of mutations that happen. Most of those mutations are detrimental. Those will make virions that will never propagate. But if one has an advantage, it's better able to attach to a cell or to spread, that's going to take over in the population. And that certainly is what has been happening since the recognition of the first SARS-CoV-2 strain and all the new variants that we've had over the past years. And so the example that I'm showing you here, so some of you might remember way back when all of a sudden we went from the original strain to Delta. The reason it was different is because it had mutations that changed sequences in the viral genome. This is the spike protein, so that protein that antibodies are made against. What it's in red here are amino acids that are different in Delta from the original strain. Shortly after Delta, we had this Omicron, which somehow picked up a whole bunch of different mutations. And that's why Omicron was so different from Delta, and why it was, people who had some immunity to this were having a harder time dealing with Omicron. And that is what has happened over the past years, is each time a virus picks up a mutation that makes it a little bit more infective, that's going to take over, and you have these variants taking over. And, this is something that just continues to happen. You probably saw in the news a few days ago, we have this XFG, it's called Stratus COVID strain, and many of you, maybe at the end of summer or recently, have had COVID, and it has probably been this one. The good news is it is an Omicron descendant, so it still looks enough like Omicron at some of its antigens that the WHO thinks that our current vaccine still should be pretty good against this variant. But this is an example of evolution and survival of the fittest being able to propagate on. All right, so let's do some basic review of the molecules and processes we're going to be talking about in this class. So we're going to start off with DNA, and the process of DNA being copied into more DNA is called replication, yes. And when your DNA has the genes, and when a gene is turned on and makes a copy into RNA, that process is called transcription, right? And so when that RNA, if it's a messenger RNA, needs to get decoded into a protein, what is that called? Translation. So those are the three molecules. Those are the three processes we're going to cover in this class. But of course, it's not that simple because there's a whole lot of regulation that goes on to determine when are you going to transcribe a gene, how much are you going to transcribe, how well is it going to be translated, how stable is that RNA going to be. And so we're going to be talking a lot in this class about the processes, get the foundational knowledge, and then examples of regulation of these processes. All right, so we all believe that DNA is the genetic material, but how do we know that? How do we know that DNA is the genetic material? And in fact, this might seem like, why are you asking this? But just 100 years ago, it was not known that DNA was the genetic material. And that's not that long ago, if you think about it. And so, in the 1920s, back in those days, people were able to identify molecules in cells, and they saw cells, were composed of a lot of protein and a lot of DNA. All right, so those are our two main contenders for being the genetic material, what's inherited to the progeny. And if you're thinking about, well, what could give all this diversity of life, you might think, well, proteins are made of 20 different amino acids. They can be all sorts of sizes and shapes and fold into all these structures. And so that's probably what's responsible for all this diversity of life, right? Especially if the alternative is DNA. DNA is boring. It's just 4 different nucleotides and sort of this regular structure. How can that give rise to all the diversity in life? So just like anything we know in science, we got there because of experiments, right? So experiments needed to be done to test these two alternative hypotheses. DNA is the genetic material or protein is the genetic material. And one of the famous experiments that helped us understand that DNA is the genetic material used these two sort of amazing organisms. And so does anyone know what this big sun-like looking thing is and these little aliens that are attaching to it? So it's a virus? Yeah, so these are viruses. What kind of cell are these particular viruses attacking? Bacteria. So this is a big bacteria cell, and these little aliens are bacteriophage. Bacteriophage is just another name for a virus. And what's amazing about these phage is that they themselves don't enter the cell. It looks like these are inside the cell, but they're not. They're just on top of the cell from this picture. So the virus themselves don't enter the cell, but they put something in the cell to make more of themselves. Question? Ah, yes. And so Alfred Hershey and Martha Chase used this knowledge that there were these phage that could attach to the outside of a bacteria cell, inject in their genetic material, whatever that was, to make more of themselves. But the phage themselves never entered the cell. So they set up this experiment, Hershey-Chase experiment, sometimes also called the wearing blender experiment, like a blender like you use to make a margarita. So we'll see why that is in a moment. And the whole key to this experiment is they used differential labeling to be able to track the protein versus the DNA. So what they did is they grew up a whole bunch of these phage using S35, sulfur 35. This is a radioactive form of sulfur. Is there sulfur in protein or in DNA or both? Protein, pretty exclusively protein. And why? What's in protein that has sulfur? Methionine and cysteine, yes. So there are 2 amino acids that have sulfur in them. And so this is a great way to just label the protein of this phage. On the other hand, they grew up phage in phosphorus 32, a radioactive form of phosphorus. Is there a lot of phosphorus in DNA or protein? DNA, right? Every nucleotide is attached together through a phosphodiester bond, as we'll see later in class. So lots of phosphate in DNA, little bit of phosphate sometimes in protein if there's a modification, but by and large, if you're getting a lot of label, it's coming from DNA. And then they just took these 2 differently labeled populations of phage, mixed them with the bacteria, allowed the phage to attach to the bacteria, and then inject their genetic material into the cell. And then they used the blender to whirl them around, and what that did is it knocked the phage off of the bacteria so they could separate the phage from the bacteria. And then they looked, is this bacteria that has accumulated the genetic material, is it radioactive or not? And they only got radioactivity when the phage had been labeled with P32. And so this is one of the definitive experiments to show that the genetic material was indeed DNA. Any questions? Okay, question for you. Let's see how well we understand about DNA being the genetic material. So get out your clicker. Use that BC. So Scrapie, many of you have probably heard of Scrapie before. It's terrible. It's an infectious brain disease in sheep. It's related to diseases that humans can get also, quite similar. And so researchers isolated the brain extracts from 6 sheep and injected them into the brains of hamsters to figure out what was causing this Scrapie's disease, what was causing this to infect fellow sheep. And so what they did in their experiment is they took one sample and the whole brain extract, and so injected that into a sheep, or into the hamster in this case. Another set they treated with RNase, it's still infectious. They treated with DNA, still infectious. They treated with proteinase, it's no longer infectious. So based on this experiment, which molecule is the genetic material? And feel free to talk to your neighbors, debate among yourselves. That's totally cool. All right, 10 more seconds. All right, put your final answer in. All right, so we have a lot of people liking our answer C, protein. Thought we just learned DNA is the genetic material. We do have a lot of votes for DNA, not so much for RNA or everything. Oh, we have a couple jokesters, huh? You forgot your clicker, but still clicked in. Okay. All right, so who is feeling confident with their answer and wants to explain their rationale for it? Yes?
I chose C, but it says when it's treated with proteinase, which is an enzyme that breaks down proteins, it's no longer infectious, so it leads me to believe that
Yes, so based on the data from this experiment, you have to conclude that protein is what is propagating the phenotype, what is causing the disease. So let's make sure everyone understands this. So when we say it's proteinase treated, as he said, that means you degraded the protein. So all the protein's gone in the sample, and now it's no longer infectious. When we treated with DNase, so what does DNase do? Degrades all the DNA. There is no DNA in the sample, but it's still infectious. RNase degraded all the RNA, still infectious. Of course, the whole untreated brain, we would expect that's our control, that is infectious. This is a real experiment that was done. It was shocking that this was the result, right? Because DNA is the genetic material. It is what like causes phenotypes. But it turns out in this particular case, these Scrapie's proteins are able to induce conformational changes of other Scrapie's proteins to cause these aggregates in the neurons that then kill the neurons and cause these diseases. And so this wasn't, I didn't ask this question to confuse you and make you think, oh no, DNA is not always the genetic material. Instead, what I wanted to do is give you an example of how there's always surprises in science, and you need to do the experiment and think about what the experiment is telling you, because maybe you'll be discovering something completely unthought of before and have to think outside the box to understand it. Do I have a question? Yeah, Prions is, yeah, another name for, yes, so Prions, Scrapie's proteins, yes. All right, is anyone confused about this anymore? No, good. So normally DNA is a genetic material unless we do an experiment and it tells us something else. All right, so let's talk a little bit and make sure we understand what the structure of DNA or RNA looks like. So DNA and RNA are just chains of nucleic acid that are attached together. They are composed of nucleotides. So when we're thinking about a nucleotide, there are 3 parts we always want to consider. So the first thing that's listed here is if it's a nucleotide, there will be a phosphate group. If there is 1 phosphate, it is a monophosphate. If there's 2, it's diphosphate, 3, triphosphate. And that'll be attached to the 5' carbon of this ring structure. That's a ribose. Sometimes it's called a sugar or a pentose because of the 5 sides to it. Is this particular ribose going to be found in RNA or DNA? Why RNA? I heard RNA. That is correct. Why? Yes, so when we're looking at DNA versus RNA, it all depends on what is attached to this 2' here. If there is a hydroxyl, then it is a ribose. If that hydroxyl is missing, if the oxygen, the OH is missing, you just have a hydrogen, then it is deoxy. Then it would be DNA. And you'll see an example of that on the next slide. So you want to identify if your ribose is RNA, a ribose, or deoxyribose. And then the only flavor these have is dependent on what base is attached on the 1' carbon. In this case, it is an adenine base. So the 3 parts of the nucleotide are going to be the phosphate, the ribose or deoxyribose, and whatever base is attached. So there's 2 main differences you're going to see between RNA versus DNA. We already identified that first one, and that's going to be whether on that 2' position you have a hydroxyl, an OH, like we saw on the last slide. It's going to be found in RNA. If it is just a hydrogen, we have removed that oxygen, so deoxy, 2' deoxyribose, that's what you're going to find in DNA. In RNA, you also have 4 different bases. You have an A. What is an A? Adenine. G? Guanine. C? And U? Uracil. So uracil is also going to be the difference from DNA. DNA has those first 3 the same, but then instead of uracil, it has a T. What is that? Thymine, exactly. So if you want to identify whether a nucleotide or a strand of nucleic acid is RNA or DNA, does it have a hydroxyl or just an H? And does it have uracil versus thymine? All right, so here are the 5 different bases and what their structures look like. You will not have to draw these on an exam, but you will have to recognize them. You will have to know if it's a purine or pyrimidine. The purines, the commonality is they are 2 rings. The pyrimidines are just a single ring. I also have some atoms circled on some of these. Why do I have those circled? Right, they are involved in the base pairing between these certain bases. What is adenine base pair to? Thymine, I even have it color-coded, right? So how many hydrogen bonds are going to be between A and T? 2. That must mean guanine base pairs with cytosine, 3 different hydrogen bonds together there. So you want to be able to recognize these nucleotides. You want to know if it's a purine or pyrimidine. And you also want to know the atoms that are going to be involved in hydrogen bonding to base pair to the partner. All right, so here's a table from your book that has all the very specific naming of the nucleotides. We're not going to get hung up with whether you tell me an A is adenosine or adenine or adenylate. That's like not so important to me. But certainly if it is a cytosine, don't tell me it's cysteine. Why? Because cysteine is an amino acid. It is completely different from cytosine, okay? So be a little bit specific. And then here's an example of the naming convention. So, okay, so is this DNA or RNA? DNA, right, because we don't have a hydroxyl on that 2' position. So if you were to name this nucleotide, you could start off by looking, all right, is it DNA or RNA? If it's DNA, it's going to be deoxy, so I'll name it deoxy. And then what base is it attached to? So this is adenosine, right? Is adenosine a purine or a pyrimidine? Purine, 2 rings, very good. And then the last thing I want to do in my naming is take it into account how many phosphates. If there's 1, it's monophosphate. If there's 2, it's diphosphate. If there's 3, it is triphosphate. If it is RNA and has a hydroxyl here and you need to name it, either just don't say anything, don't say deoxy, or you can say ribose and then name the rest of it. So whenever you're naming a nucleotide, think about those 3 parts and make sure everyone is accounted for. All right, the other thing about DNA, especially when it's base paired, is that there's going to be directionality to it. There's always going to be a 5' end and a 3' end, and that just refers to that carbon. So this is the 5' end that has that 5' carbon versus the 3' carbon that's part of the ring here, and that's going to be attached to this hydroxyl at the end. Whenever you are writing nucleic acid sequences, like eventually when you're going to design primers for PCR, always, always, always write the 5' and the 3'. That will make it clear what sequence you mean, and it'll really help make sure you don't get confused with which way base pairing is going. So given that advice, if I asked you what sequence is going to base pair to 5' CAG 3', what would you write first? 3, so you can write a 3', and then what are you going to tell me? G, T, C, 5', exactly. So you know your base pairing rules, write the right letters, and then make sure that you always have this directionality, and that is what we call anti-parallel. If this is the 5' end here, it's going to be 3' end down here. All right, so let's talk then a little bit about the 3-dimensional structure of DNA. So when I show you this beautiful picture here, what are some descriptors that come to mind? Helix. It's a helix. Anything else? What's the helix composed of? Yeah? Yeah, double-stranded, 2 strands that are wrapped around each other. Yeah, so we have a helix, it's double-stranded. How do we know that? Experiments, right? Experiments needed to be done to solve the structure of DNA. And the most important experiment that was done that led us to this beautiful helical structure was performed by Rosalind Franklin, her X-ray diffraction data. So the way X-ray diffraction works at a very simple level is you take the molecule you're interested in this case DNA, you have a very concentrated solution of it, and you pull into very, very thin fibers. You shoot X-rays at it. They diffract in a particular pattern that is captured, and then you use very complicated math to solve what structure that pattern is telling you. And so when she got this beautiful result, what she could conclude immediately is that DNA is a helix, and it's at least 2 strands. All right, so at this time, people knew what a strand of DNA looked like, and so if you know that it's a helix and there's 2 strands of it, you can start building models and thinking about what that structure might be. And so this is just a thought experiment. Don't worry about copying this all down. I'm just showing you a strand of DNA and the same strand of DNA, because we know we want 2 strands, and we want them to come together in a nice helix. How about let's practice our bases first. Which base is this with this methyl group? Thymine, exactly. So thymine has the methyl group, this CH3. So that's thymine. All right, is that purine, pyrimidine? It's a pyrimidine. So this one must also be a pyrimidine. Which one is this that looks like it can make 3 base pairs? Yeah, C, cytosine. And then this must be a purine. Looks like it only base pair twice, 2 nibs. Which one? Adenine, yes, exactly. So, all right, so we have our DNA here. It is held together by phosphodiester bonds. And we have 2 strands, and we want them to come together into a nice helix. And so this is actually how Watson and Crick actually started off in trying to think about, build the model for DNA. They built physical structures and thought, how can these strands come together? And so when you're thinking about DNA, again, we know it's a genetic material, but it's kind of boring. The only thing that's interesting are the bases. And so that's the only thing that maybe like proteins can recognize as different. So those bases should be on the outside, right? So the helix should come together with the phosphates in the center and the bases on the outside so that it can be read by proteins. Is that correct? I see you shaking your head no. Why? But why? What happens if we do it the other way? What if the bases are on the outside? So if you have phosphate next to phosphate, what happens? Boom, they're going to blow each other apart, right? Yeah, you don't want negative next to negative. There's no way that would hold together in a strand. Basic chemistry. That was the first model that Watson and Crick came up with. It was the phosphates on the inside, bases on the outside. They thought they'd solved it. They presented this to Franklin and some other people. Got totally ridiculed because that breaks all the principles of basic chemistry. You weren't even like thinking about like how molecules need to come together. And so, of course, that was embarrassing and humiliating. But what did they do? They didn't give up and like, oh, we'll never solve this. They just moved on. Let's build another model instead. And then, of course, the next model is what turned out to be the correct one and what won them the Nobel Prize. So if you make a mistake, especially a mistake that everyone like knew the answer to, just move on. It's not a big deal. All of us have done that in the past. All of these geniuses and greats have also made simple mistakes and moved on. All right, so we're going to put the bases in the center. Okay, so now we've moved the bases in the center here. And we're going to want them to come together. But they're sort of far apart. So what else do we need to do to allow these strands to come closer together? Yes? So we're going to want them to hydrogen bond, but they're still like far apart, right? Yes? Cross them over. What do we need to do? Make them, if this is 5' to 3', invert it, make it anti-parallel, exactly. We just need to flip that over. So now we have 5' up here and 3' here, 3', 5'. And now you can see how these bases are going to be able to be brought close together for hydrogen bonding. These 2 particular pieces of DNA that I've drawn here, are they going to perfectly base pair to each other? No. Where's the problem? So we have A and T will base pair, A and T will base pair, C and C. No. So these would not perfectly base pair. But it was that type of model building that then led to sort of this idea of how you can bring these strands together in a nice helix. But of course, the other key, and it would like only it was made possible, again, by experiments, was understanding the base pairing rules, to knowing which of these bases compare with each other. And so some of you may have heard this as Chargaff's rules, and that's because he did the experiment to come to the conclusion that A will base pair with T and G will base pair with C. So what did he do? So he took DNA from a number of different sources, chopped up that DNA into individual nucleotides, identified the percent of that nucleotide in the sample. And so if we look at our first example here, this sample 1 from sperm, what you see is the total. So recovery, you never get 100% when you're doing a real experiment, right? So, but you got pretty high here. There is pretty similar percent of A and T and a perfectly same percent of G and C. That's interesting. And then when you do it with all these different samples, it always keeps coming out that way. Why? Every time you have an A, you have a what? A T. Every time you have a G, you have a C, exactly. And so this is what led to this idea that the way you would have the same number of those is if they were paired to each other in a double strand of DNA. And so it was that recognition by Watson and Crick that allowed them to come up with this model of the double helix consisting of the antiparallel strands and that the base pairing rules would allow those two strands to be held together. And so they, along with Wilkins, were awarded the Nobel Prize in 1962 for this modeling, right? They never actually did any experiments themselves. They just used other people's data and came up with the original model that proved to be correct. Why did Wilkins get the Nobel Prize? Why didn't Franklin get it? She provided the first data. Does anyone know? It's so sad. So Franklin died, actually, before the Nobel Prize was given, and you're not eligible once you die. Wilkins was actually her boss, and after they proposed this structure of DNA, he actually performed a lot of experiments after that to sort of validate that this was likely the structure of DNA. All right, so let's look a little bit closer at this structure of DNA as those two strands wind around each other in a helix. In one full turn, there's about 10 base pairs of DNA. This is a picture here of the base pairing between A and T, which atoms are involved in A and T base pairing to make 2 hydrogen bonds, and then G and C to make these 3 hydrogen bonds. Again, we have antiparallel, 5' to 3', 5' to 3' to allow those strands to come together and have base pairing. Now, those grooves, go back a second. So one of the confounding things in the beginning and what led to Watson and Crick having the wrong structure of DNA is how can a protein tell what the sequence is of DNA if it's hidden in the center? But the nice thing about this helical structure is it's not entirely hidden, right? Those edges of the base atoms are accessible to proteins to recognize. And there's a major groove where there's more accessibility, and then the minor groove where it's a little bit more compact. But these atoms at the edges of the bases are then accessible so that proteins can read or can interact with those atoms in the bases to have sequence-specific DNA binding. And so here's an example of this white sort of loop structure here. This is a protein structure. It's a protein called the Tata box binding protein. We'll talk a lot about that when we talk about transcription. It will recognize specific sequence. In fact, the sequence it recognizes is TATA. That's why it's called a Tata box binding protein. And it will bind to that specific sequence. And it will cause bending of the DNA, so it can actually alter the structure of DNA. And that can be important for allowing additional proteins to recognize additional sequences in the DNA, and in this case, to set up the pre-initiation complex to start transcription. All right, so for DNA to actually be useful in our cells or in the laboratory, we often need to separate those two strands so that they can be copied. And so what kind of base pairs are holding our two strands together? Hydrogen bonds are what's holding them together. That's what this base pairing is. It's hydrogen bonds. Those are the types of bonds that are connecting the two DNA strands. And so in the laboratory, if we want to denature DNA and make it take double strand, make it single strand, how do we do it in the laboratory typically? You heat it up, right? Hydrogen bonds they're not covalent bonds, they are semi-weak, and so if you heat them up, you can blow apart those hydrogen bonds pretty easily. But in your cell, every time your DNA wants to replicate or be transcribed, it needs to also unwind or be denatured in order to read the sequence. Does your cell heat up to boiling every time that it needs to be copied? No. What happens in your cells? What unwinds DNA in your cells? Helicase. There's a type of enzyme called a helicase that can unwind this double strand, can break those hydrogen bonds, and we'll talk about that when we get to DNA replication. So that's how we can denature DNA. In a cell, we use helicases. In the laboratory, we use high heat. Sometimes we can also use chemicals or change the pH. There are a variety of things that will break hydrogen bonds. All right, so we need to understand what this term means. So TM is short for melting temperature. The definition of melting temperature is simply the temperature at which half the bases of double-stranded DNA have been unwound, have been denatured, are single-stranded. So let's look at an example of how we can determine the TM for a sample of DNA and whether it is double-stranded or denatured. So in the laboratory, we can use 260 nanometer light to take an absorption of a sample of DNA. The bases on DNA, their electronic structure, can absorb light at 260 nanometers, but only if they are not base paired. If they are base paired, they cannot absorb light the same way. So when you have double-stranded DNA and all those bases are already base paired to themselves, you have much lower absorption than if they are single-stranded, and then you can absorb high amounts of this light. If you look at this graph here, what you'll see is if you have a sample of double-stranded DNA and you change the temperature, you go from double-stranded to single-stranded. Makes sense. We just said that in the laboratory, if we boil our DNA, it goes from double-stranded to single-stranded. So to find the melting temperature of your sample of DNA, if you take the absorption and you find that the halfway point, sort of between 0.5 and 1, is going to be 0.75, and you just go out to your curve where you're absorbing, where you're detecting that DNA, and look at the temperature at which your sample is half denatured, that's your melting temperature. So in this particular example, it's about 83 degrees Celsius. That's what we mean by the melting temperature. So here's a question for you. So what would happen to the absorption if you took the denatured DNA and slowly chilled it on ice? What would happen to the absorption? Go down, right? You just follow it down. As it became more double-stranded, it would absorb less light. The absorption goes down. Okay, so we can use this property to estimate the percentage of GC in double-stranded DNA. And it turns out that the higher the GC content, the higher the melting temperature. Why is that? Sure. Yeah, GC is how many bonds? 3 versus AT? 2. It's going to take more energy to break 3 bonds than it is for 2 bonds. And so that's exactly what we see if we look at this graph over here. So if the melting temperature is, let's say, 80 degrees, we see that this is going to tell us, based on experiments that were done here, that the percentage of GC, just go up and over, is going to be about 20% GC. How much cytosine do we have in that sample? What percent? 10%, exactly. So we went from 80, I think I have a graph here, right. So if I tell you that melting temperature is 80, all you have to do in the graph is go up to where the line intersects, look at the percent GC pairs. So you have your sample has 20% GC. You know that you have an equivalent number of G as C, so half of 20% is going to be 10%. Does everyone see where we got that? How much A do we have in that sample? 40, right, because if we're 20% GC, we must be 80% AT. Half of 80% is 40. Is that clear to everyone? Good. Here's a question. If two samples of genomic DNA have very different TMs, let's say one is 80 and one is 90, can they be from the same organism? Same species, same as you and me. We have the same TM. Would we be different by 10 degrees? No, we would definitely not be the same species anymore. Why? Because what does that mean if you are that different in melting temperature? Your percent GC or your percent AT is wildly different, right? And species have essentially the same DNA sequence. How about the reverse, though? Can two organisms have the same TM but be very different species? Yeah, absolutely. So here's an example of that. So here's an example of the TM for a variety of different organisms. And oh, look, we see a cow and salmon have the same TM. All that means is their percent GC is pretty much the same. But then how can they be such different organisms? What is different? The sequence that GC comes in, exactly. So you can have the same percent of nucleotides, but they can be scrambled in a totally different order to make a different organism. Is that clear to everyone? All right, that is the last piece of information we will go through tonight. So just some reminders, make sure to submit your pledge and register your iClicker if you have not. Have a great weekend.